reticulum, so then these complexes protect the viral genome and the virus as it's replicating, because this is RNA, and so you'll get sensed, especially when it forms the double-stranded form, the minus sense strand. Anyway, so then you see this kind of clustering forming, and then you start to get synthesis of the so-called subgenomic RNAs, and these messenger RNAs, and these encode for the structural proteins, right, the spike and so on, right? And so then you finally, and this is all happening here, and then finally you start to get assembly of the different viral proteins that has to come together with the genome, which has been replicated, right? So then you get production, synthesis of the viable infectious virus. So I'm kind of mentioning this because there are several stages along this pathway where you can get an outcome that's not what you started off with. You don't get true replication of the parent strain, right? So this virus has mechanisms to make sure that the replication is stringent. Of course, this can fail, but also mechanisms that then allow the diversification that we're seeing, and then other mechanisms which then support production of viruses which are already primed so that they are much more infectious when they infect the next cell. And that's to do with the furin proteases in the host cell, and also the way that the virus is primed at the surface of the cell. So there's many of these, and there's been quite a lot of work in the last year or so to really kind of come down on what we understand, how the virus replicates. One very interesting thing is that the virus doesn't just enter the cells in this way, by fusing to ACE2 and then being primed by the tempress, by the surface protease. It does also enter the cell via the way that many other viruses do, which is just by receptor-mediated endocytosis. So it can just enter the cell, and that means it doesn't have to be primed by the surface protease, and it doesn't have to bind so stringently to ACE2, because there's different, and that's part of the reason why you get this differential replication efficiency in different parts of the respiratory tract, right? Because the distribution of those molecules and so on. So that's what we're coming to understand now, and that's particularly in the case of Omicron, by the way. That's what Omicron turns out it can do. So I'm just mentioning all that, a brief summary, but that's a lot of work's been done in the past year to show that. And also this part here is the way that translation of the subgenomic mRNAs occurs, and the transcription is really what we understand is so important now in evolution of these recombinant viruses that we're seeing, right, in real life. So we're seeing it not just in vitro, but it's being measured now in transmissions in various places. All right, so just going back to this point that when this virus first emerged, some of us, including me, sat around and said, oh, this is going to be fine, because this virus is a coronavirus, very large, has a very large RNA genome, and it has to have this, it does have this proofreading capacity where as replication is occurring, this is NSP14, which is an exonuclease, right? So it proofreads as the replication's occurring, right? So coronaviruses have this because their genomes are too large, right? And otherwise they wouldn't be able to replicate. So I thought, well, that's all going to be fine. It turns out not so much. It probably prevents a little bit of antigenic drift, just kind of substitution errors, right? But not recombination. In fact, it turns out NSP14 actually supports recombination, right? So that's just, I'm mentioning that because that's important. I think this is the lab. Okay, so, and again, I'm just mentioning this again. So this is what's happening. This is the plus sense, and then this is the minus sense that needs to be synthesized off it, and then the subgenomic mRNAs. So you have this discontinuous transcription here during the synthesis of the negative strand. And so what you get is the polymerase jumps along the genome, right? So as it jumps like this, it can misincorporate the different strands, the body strands, right? And stick them up with the leader at the top. So then you get these subgenomic mRNAs that actually are chimeric, right? And so there's been description of that in transmissions among people. So to do that, you have to be infected with, not in this case, but you can also be infected with more than one variant, if you like, and that also supports this. But this is the other mechanism that also occurs. All right, so after all that, we're seeing now emergence of all of these different variants. And we've seen it happen in real time. We've lived with it the last couple of years. And so this is showing you the kind of a little diagram here, Venn diagram, of the similarities among these different variants. I think this font is a bit too tiny, but the concept here is you end up with Omicron, which has a huge number of mutations, substitutions, amino acid substitutions. Especially on the spike, which is the receptor binding domain also, which is the part that attaches to the host cell receptor, and it's the part that can evade immunity, because you'll get a replication despite that. Okay, so this is again just showing the magnitude of transmission that we've had in the last couple of years, and then this is what's happening now, especially in Africa. Not pointing out Africa, but saying there's a lot of data coming out of Africa. And again, emergence of Omicron, but I've included this slide to show, just to illustrate how distant Omicron is from all the other variants of concern that have been circulating. Distant meaning genetically, but also phenotypically, antigenically distinct, right? So that's why we're seeing all this escape from vaccine and also from even detection in the first place, right? So that's why we're seeing all of this. So, but the fact that you've got this emergence of this very distinct virus, the question is, of course, how did this actually happen, right? So there's a lot of chatter about that. I don't think anyone's really saying anything about lab origins, which is kind of weird. You'd think they would be by now. So this is the three main hypotheses circulating at the moment. It could have circulated and evolved in a population with little surveillance and sequencing. Right? So it's just got out there somewhere, right? And people like to say, oh, it must be happening in Africa, right? Because lots of countries without that much. But then some people think, well, in fact, most places in the world are probably being monitored to some extent. So that's maybe not so likely, but who knows, right? Or it could have evolved separately in a chronically infected patient. And we've seen examples of that very clearly. The question is that significant enough to then seed like all of the viruses going forward, right? That's the other unknown. And this is an interesting one. It could have evolved in a non-human species from which it recently spilled back into humans, right? So that's a human to animal and then back to human, right? So that's possible. But the question is that really that significant, right? And you can see descriptions of that, but I don't think it's anything to get scared about and start sacrificing your dogs or something, but certainly that's possible. So I'll just show you an example of this. This is a recent report. This is a phylogenetic analysis of 7 genomes, whole genome sequences from a patient with advanced HIV infection in South Africa, right? So this is showing exactly what will happen. These are the local sequences, and then this is the variant that evolved in this person co-infected with HIV, who had HIV infection and then was infected with COVID-19. And they picked this up because this woman became sick, and they were following her up, and then the sequencing showed this. So she was on HIV treatment, antiretroviral therapies, and not responding so well. She had resistant viruses, so she was getting replication still. And then this is the same time. So, and then also she was obviously immune deficient. But then once she was switched to then control her HIV infection, in other words, allowed some reconstitution of her CD4s and immunity in general, then you could see that her capacity to control SARS-CoV-2 replication came along with it, also occurred. So it was a reconstitution of her immune capacity, kind of was associated with this clearance of SARS-CoV-2. But in the meantime, there was this evolution, because she was viremic for about 6 months, as opposed to the few days when we expect to be viremic. This was ongoing. And so you get this immune pressure, even though she had lower CD4s and lower immunity and so on, there was still enough to drive this evolution. So you can see it, right? And there's been several other cases also, not HIV co-infection, but certainly people being immunosuppressed for cancer treatment and so on. I didn't show you all those examples, but there's a few, and that's very clear. So that was also the first idea for where alpha came from, and maybe even delta, and certainly Omicron, right? Was it something like this, or could it be that and a recombination with some other species? Who knows, right? So we just don't know. But certainly this kind of diversification is not just possible, we see it all the time now. And the work in the last couple of years, especially the last few months, has really shown that. And I'm just showing you this very boring kind of text because I didn't have a pretty picture to go with it, but this is, we can see that we can capture this intra-host recombination of SARS-2. This is during a superinfection, meaning this person was infected with more than one virus variant. You can measure it with alpha and epsilon variants in New York City. And this is evidence of recombination among circulating viruses, right? So you could see this and map this out, and you can see here there's widespread kind of recombination within the virus that this person ended up, that was dominant in this person. So I'm just saying that so we can measure this, right? All right, so coming back to Omicron, because this is what we're living with basically now. And this is a nice diagram, a nice article that came out in New Scientist actually, but summarized very nicely some of these studies that have been done in the last year, right? So just making some nice pictures, also good. So this is showing basically data to strengthen this idea of why Omicron is so much more transmissible than the other variants have been. So when this started to happen, some of us discussed it and thought, well, the immune background is very different, right? So how do we separate those two things out? Is it an inherent capacity, property of the virus itself, or is it the virus on the background of much different, a different immune background to when we first started off, right? And so all of the above, really. So this is showing, of course, the extreme diversification of the spike, especially the receptor binding domain on the spike protein. And then this is now showing these other interesting mutations. This variant has more than 50 substitutions all along its genome, and many of them, 20-something, on the spike itself, right? That's a lot. That's very diverse, right? So what this is showing, though, this work that's continuing, so not just focusing just on the spike, but looking at, not just on the RBD, the binding domain, but looking at the rest of it, you can see that these other mutations also contribute to the phenotype of the virus, the way it infects the cell, the way it replicates, and its stability and its transmission capacity, right? So there's a lot of interesting work being done, but basically you have this kind of stabilization of the spike, because of course in the early stages of the infection replication cycle, you get this priming event and you get separation of the. Okay, so yes, so these other substitutions apart from the ones in the RBD serve to really stabilize this virus, make it much more infectious and much more transmissible, right? So there's a lot of work that's gone into that. I'll just say, here's a pretty picture that summarizes all of that, but there's been a lot. And then finally, this other interesting alternate cellular entry, especially by not having to rely on the capacity of the spike to recognize and bind strongly to ACE2 or to be cleaved by temperus, or for temperus to be there in the correct form and in the correct concentration and all that, because there's the distribution of these molecules not equivalent, right, throughout the respiratory tract. That's the other thing. So it's also able to then enter by receptor-mediated endocytosis, which is one of the likely or possible reasons why this virus appears to replicate so much more efficiently in the upper respiratory tract versus the lower, and so therefore traditionally those viruses that do that are less virulent, right? Less associated with more severe disease. This is not a mild virus. You don't want to get this just to, why not? But this is the fortunate kind of situation. Okay, so just going on a bit more about diversity and neutralization, because this is important for understanding how the vaccines will work, or have worked, which is what I'll talk about next. But this is a really nice paper that I think it's still in the review part. But this is looking at antigenicity again, right? Antigenicity of Omicron compared to the other VOX, the other variants of concern. So it's very diverse. Here it is, right? Very diverse. And then so basically you can see this evolution of these different forms, which have basically been selected for different reasons, but now we're ending up with Omicron, which is quite distinct and which has escaped, can escape, we think it's escaping, the immunity that's been conferred by the previous viruses, right? Because we don't live in that environment, but in other countries, people are living in environments where they've